Good day, we are group 13 and for our concept assignment we chose the process DNA replication. DNA replication is one of the most basic processes that occur within the cell. It is the biological process of producing two identical replicas of DNA from one original DNA molecule that acts as templates. This way, the daughter strand contains exactly the same genetic information or DNA as the parent cell. DNA replication occurs in all living organisms, acting as an essential part for biological inheritance. The replication of DNA is semi-conservative. This means each strand in the DNA double helix acts as a template for the new complementary strand. There are three stages of DNA replication, which are initiation, elongation, and termination.
The first step is initiation. Replication always begins at a particular location known as the origin of replication. This is a sequence of DNA at which replication is initiated. The enzyme DNA helicase aids with the unwinding of the double helix of DNA strand by breaking the hydrogen bonds holding the complementary nucleotide bases adenine, thymine, guanine, and cytosine of DNA together, so that the two strands can be used as templates. The two strands unzip, form a Y shape called replication fork. After the strands are separated, we have a leading strand, which is synthesized in the 5' to 3' direction towards the replication fork, and the lagging strand, which is synthesized from 5' to 3' direction away from the replication fork. This means the daughter strands are synthesized through different methods. One, the leading strand aiding the nucleotides one by one in the direction of the replication fork, and the other of the lagging strand aiding nucleotides in chunks. DNA single-stranded binding proteins bind to the DNA to stabilize the single-stranded structure generated by the action of the helicase. Replication is 100 times faster when proteins are attached to the single-stranded DNA.
Next is the lagging strand. DNA polymerase on the leading strand can only follow the replication fork because DNA polymerase must move in the 5 to 3 direction on the lagging strand. The enzyme must move away from the fork. The lagging strand begins replication by binding with multiple primers. Each primer is only several bases apart. Another DNA polymerase then adds pieces of DNA called Okazaki fragments to the strands between primers on the ends of each RNA primer. Each individual segment is replicated away from the replication fork. This process of replication is discontinuous as the newly created fragments are disjointed. Afterwards, the nucleotide replacement enzyme removes the primers of the lagging strand synthesis. The next step is elongation. For the leading strand, an enzyme primase produces a short piece of RNA, a primer that binds at the starting point of replication, the end of the leading strand. the primase works to where replication can begin. Through the process of elongation, an enzyme called DNA polymerases creates a new strand. DNA replication is inflexible, meaning this enzyme only functions in the 5 to 3 direction. DNA polymerase binds to the leading strand and sits near the replication fork, moving as the fork does. It starts by adding the complementary nucleotide base accordingly. Since DNA replication moves along the parent strand in the 5 to 3 direction, replication occurs easily on the leading strand. As a result, the nucleotides are added in a 5 to 3 direction. Now a nucleotide replacement enzyme is needed to remove the initial RNA primer of the leading strand synthesis.
The last step in replication is termination. Once the process of elongation occurs and there is no template strand left to replicate, replication is terminated. An enzyme called DNA ligase joins Ozaki fragments together, forming phosphodiester bonds between the nucleotides, creating complete strands. Synthesis is now complete and the new strands are bound and stabilized. You now have two identical double helix DNA molecules from the one original double helix DNA molecule. They are semi-conservative because the two copies each contain one old original strand and one newly made one.
For our model, we use different colors of cardboard to represent nucleotides. Pink representing thymine, orange as adenine, guanine as green, and cytosine as blue, while the deoxyribose is presented by a yellow chain.